Welcome everybody. It's a great pleasure for me to present you some initial findings of our consortium called ARREAL, or the Aggressive Infection-Related East African Lymphoma Initiative. The broad aim of this program is to improve the diagnosis of EBV-driven lymphomas in East Africa. You probably know the basic facts. There are different types of EBV-driven lymphomas. Hodgkin's disease, diffuse large B-cell lymphoma, some T-cell lymphomas can be EBV-driven. But endemic Burkitt's lymphomas are no doubt the most famous lymphoma that is EBV-driven, and it occurs primarily in sub-Saharan Africa. It's actually the most common childhood lymphoma worldwide because lymphomas are otherwise exceedingly rare in children. It is defined by the presence of the MYC translocation, whereby MYC translocates to the either heavy or light chains of the immunoglobulin locus. Malaria has been identified as a cofactor in the pathogenesis. It can occur in endemic forms, sporadic forms, or also as an HIV complication. The epidemiology of Burkitt's lymphoma is really an object of ongoing research, and I feel very privileged that the ARREAL program has teamed up with a previous initiative called Emblem, and this was a research consortium in Uganda, Tanzania and Kenya, and was really aimed to further defining epidemiology of Burkitt's lymphoma around the great East African lakes. So what this consortium identified, amongst many other results, is that children with Burkitt's lymphoma do indeed occur more frequently or live more frequently around the Great Lakes, close to the water, that they come from rural areas, and that they usually come from a lower socioeconomic background. And the presence of malaria either indirectly or directly evidenced by regular use of insecticide spray, by inpatient treatment of malaria, has been also associated with the presence of or increased presence of Burkitt's lymphoma. Burkitt's lymphoma in the children is both in endemic and sporadic forms is very well treatable. In fact, you can see with optimal treatment, event-free survival and overall survival are extremely high. Over 90% of kids survive. This has been further improved in the high-income countries where the sporadic form is more common, by the addition of rituximab to the chemotherapy backbone, as you can see on the right. However, the situation is still different in resource-restricted settings. This is an example of Kampala, where you see that roughly 40% of kids with Burkitt's lymphoma survive beyond 3 years. So the background to our program is really based on the fact that a large proportion of this poorer prognosis in resource-restricted countries comes from the fact that Burkitt's lymphoma is diagnosed so late, and the children present late, but then also once they've presented, there are delays within the diagnostic pathway. So there's a lack of awareness on the one hand, and that is not what this program addresses in the first instance, although it does indirectly. And then there is a second part, which is the hospital-induced delays, and these are primarily due to skill shortages, the technical and quality assurance issues in tissue processing of the biopsies that are required to diagnose the children, and then generally in a lack of capacity, because the whole diagnostic process is quite complicated and requires interventional surgeons, in particular for abdominal presentations, and also of course need pathologists both on the technical and also on the clinical side. So this was our goal, to develop, validate and clinically evaluate and then deliver effective diagnostic tools for the early and accurate diagnosis of infection-related lymphomas in sub-Saharan Africa. And really what we're trying to do is to compare the best local pathology against liquid biopsy. So as part of that, we have first couple of years we've been developing the liquid biopsy test that is based on sequencing of cell-free DNA, and then in the second part we'll be comparing this with the best pathology. And in order to do this in a fair way, we have spent the first 2 years improving capacity and quality of the local pathology, and this is done by introducing very advanced digital pathology, but mobile whole slide imaging amongst others, but also training the technical staff and introducing quality assurance measures into the pathway. And then in the second 2 years we perform a head-to-head comparison, whereby the primary endpoint is the time from presentation to the oncology ward to a diagnostic report, and secondary outcomes are overall survival and then a health economic analysis of those two different diagnostic pathways. So the program also supports free diagnosis and treatment, including CT scan and ultrasound scan, and then importantly we have every child gets rituximab for 4 years when they're considered for the study, because this is now the standard of care really for together with chemotherapy for diagnosis for treating kids with Burkitt's lymphoma. So the team is there, over 30 people actually involved with the program in one way or the other, but the team leaders are depicted here. They're coming from various different institutions, from Mumbili National Hospital, from KCMC up in Moshi, from Lachor, where Burkitt's lymphoma was first described in northern Uganda. Muhas is performing a large chunk of the development work and also the sequencing, and then Oxford is involved in the tech transfer and training. There's a number of different work packages, and clearly we have a clinical team looking at collecting clinical data, in particular following the introduction of rituximab, because that's the first in sub-Saharan Africa, and we need to obviously look for safety and also efficacy there and feasibility. There's a pathology work package, as already mentioned, where we've been evaluating new pathology devices, but also creating local capacity. And then there's a DNA work package that develops and then runs the liquid biopsy testing using Illumina sequencers in Tanzania. The health economics work package performs very extensive microcosting of both of those diagnostic tools and sensitivity analyses. And then we have a big training package and sustainability work package. All of this is done completely locally and by local PIs. So I would like to focus the next part of my talk really on the development and validation of the ctDNA sequencing panel, because people, there might be a perception that it's very easy to perform next generation sequencing, but of course in the nitty-gritty detail, the NGS technology has become a science in its own rights. So particular, obviously here we are dealing with liquid biopsy and cell-free DNA, which consists of very small amounts of DNA, and the data is very fragmented. So the prediction modalities that we have been using to achieve sensitivity of our analysis consists of a number of different things. So we're looking at the ctDNA quantity in each child, the fragment size of the DNA, the sticky ends of the fragments, because of the integration sides of whether they are different between the tumor and the germline cell-free DNA that you find in the plasma. We're then looking for presence or absence of EBV reads, of course, the EBV quantity. So we have developed a way of semi-quantitatively assessing the EBV read frequency, the presence and absence of MYC translocation, and then the presence or absence of other types of mutations that occur recurrently in other EBV-driven lymphomas, such as Hodgkin's lymphoma and diffuse large B-cell lymphoma. Other attributes of the test are, of course, that it has to be cheap, the libraries have to be performed easily, and it has to run on an Illumina device that can be used without having to batch samples. And so we chose the iSeq for this reason. Although we are also having MySeq capability. We have already approached over 270 patients now. This is a data cutoff from June of this year. Quite a few were excluded because they didn't meet the inclusion criteria, but we have in July enrolled 162 patients. We've obtained samples on all of them. We're also obtaining sequential samples as kids are going through their treatment. We have used the strect tube protocol for with double spin to make sure that there's no leakage of germline DNA into the plasma, because that really impacts on sensitivity. So you can see the diagnosis overall. We have 109 lymphomas by pathology, and then 53 non-lymphomas, and we need those non-lymphomas obviously in the first part of the study as our negative controls for the test validation. We've have performed several training workshops, and the Lacho and KCMC teams have been coming to the MUHAS lab to be trained. I should say that we do have a sequencing facility now also set up in Uganda, because we felt that we should use the national lab there as a disseminator of the technology from a Ministry of Health perspective. In Tanzania, we are using the lab at MUHAS at the moment. So you can see the results here of the initial result of the initial analysis. You can see that there's a big difference in the cell-free DNA concentration, as you'd expect, between healthy individuals and patients with Burkitt's lymphoma. We did also look at other types of lymphomas and also of other conditions, in particular, obviously, glandular fever, which is another EBV-driven disease. And you can see that there are increases in cell-free DNA concentration amongst other conditions, but also that there is a very nice already initial indication of that something is wrong from a cell-free DNA concentration perspective when you're looking at these data. So our design panel, there are in fact two panels. The panel A is the diagnostic panel, which contains the target genes that you can see at the bottom here. As already mentioned, in particular EBV, obviously the areas that are important for the strain definition, and then the MYC translocations. Panel B will allow us to perform whole genome sequencing of the EBV genome, because we are also interested in identifying EBV integration sites in our kids, and look at other variables, in particular substrain variables that might be associated with or not associated with Burkitt's lymphoma. And that work hasn't really been done in the field yet, so we don't really understand whether the reason why EBV occurs so frequently around the lakes, whether that is also driven by different EBV strains. We achieve very good sequencing metrics that is coming from our iSeq, with a mean coverage of 100x at 99%, at 500x it's 95%, so very, very sensitive sequencing. We have a huge number of reads, but you need those because of the cell-free issue, and the cell-free DNA is only a tiny minority of the overall DNA that you will sequence. We have a bioinformatics pipeline that is hosted by an Amazon cloud that is HIPAA compliant. It is our own cloud of the program, so we essentially we can share the data, the sequencing data in this cloud, and then analyze it together between the African teams and the UK team. The pipeline allows us to perform EBV quantification, as I've already mentioned, the SNV calling across the various different tumor-associated mutations, and then also the translocation analysis, which I'm sure everybody knows who's familiar with target sequencing is not that trivial. When it because we have to do a lot of tiling of the panel, and the MYC breakpoint is spread in endemic Burkitt's lymphoma across several megabases. So we are basically capturing not just the MYC breakpoints, but also the breakpoints within the immunoglobulin locus. And the immunoglobulin locus is a private gene, so you can imagine that this is not trivial. But we've got so the we tested some initial cell lines to make sure that at least we can detect the EBV sequences and the translocations, and you can see that in red here we can wherever it was known from the data was known from the cell line, we were able to detect it. So we then visualize, as you in line with what is recommended by diagnostic guidelines. We make sure that we visualize and visually inspect every important finding. So here you see that the MYC translocations in these patients are supported by over 100 reads, which is sort of what we are aiming for when it comes to translocations. Then we've spent some time making sure that we can precisely quantify EBV, because there's data in the literature that clearly indicates that the levels of EBV are an indicator of the presence of malignancy, and this is particularly has been studied very well by Dennis Lowe in nasopharyngeal carcinoma in China, and has led to the rollout of a qPCR test for EBV in China as a screening tool for nasopharyngeal carcinoma. So EBV is essentially located in the viral DNA is located primarily in the lymphoid cells of the peripheral blood, but then of course it is also present in the plasma, and that's sort of illustrated on this cartoon here. But so it wasn't quite clear to us, particularly for Burkitt's lymphoma, because that had never been done, whether which cell compartment we should actually use for our quantification initially, using qPCR, and it was also not quite clear in what type of sample preparation system, the cell types or the tissue types should be collected. So we ended up doing a quick comparison between plasma, buffy coat, and whole blood, both taken from EDTA and from STREC, which is this particular fixative that we use for cell-free analysis to fix the white cells so that they don't leak into the plasma. And when we then essentially performed cell-free and genomic DNA analysis on those different types of preparations. And we used both sequencing using our EBVL panel, and then we also used qPCR as the gold standard to compare against. So just briefly, the results, you can see that, as one would have hoped, we only detect the EBV. In this case, it was an NK lymphoma. The only detect EBV sequence reads in the where there's a tumor and not in the negative controls in purple, blue, and pink. So that's all good. And we did detect this EBV in different types of preparations, but clearly the cell-free STRAC plasma preparation that we were planning to use anyway is by far the best preparation to use. The other thing to say is that we've tested various different types of EBV conditions, including an infectious mononucleosis in orange, the 2 different time points of a patient going through treatment with NK cell lymphoma, and then also an endemic Burkitt lymphoma and a diffuse large B-cell lymphoma. And you can see that they are measurable levels, and that they're highest in the NK lymphoma before a treatment. So how about this basically will allow us now, we haven't got any data on this yet, sequencing data, but it will allow us to measure the EBV quantity from sequencing data. So in terms of the mutation analysis, we have got already performed some variant filtering using really the standard filters that one would use in cancer when you're looking at cell-free DNA. And you can see that quite a number of pathogenic mutations are coming up here. And there are mutations in MYC, which is well known in about 2/3 of patients, because these are close to the breakpoints of the MYC translocation. But they don't occur in all of them, so they can't be really used as a surrogate for the translocation. What we will also do, and this is just the first glimpse into the data, what we also do is obviously look at the predicted functional impact of variants. As one would expect, most of them are missense variants, and it is also known from NF-kappa BI that there are a lot of mutations in the 3' region of this gene that's been associated with regulatory impact due to microRNA that are binding in the 3' UTR. And we can also associate, when we have more data, we will associate the different incidence of mutations with potential clinical outcome. So the next step of our work will be to really start sequencing the remaining 162 patients from the retrospective cohort. That we hope that will be finished in January. We're actually at the moment installing and being trained on a MySeq here in Dar es Salaam, so that will allow us to get through the backlog quite quickly. And then we will start early next year the head-to-head prospective study that compares the pathology with the liquid biopsy as a primary endpoint. So we then perform additional analysis, and we have plans already for the EBV genome analysis. As I already said, we're particularly interested in integration sites and different substrains. And then we will explore whether we can also use Oxford nanopore technology. Now that is quite tricky because this technology is optimized for long read sequencing, and as I said at the beginning, cell-free DNA is very fragmented and it's very dilute. So it is not actually thought to be that feasible to use Oxford nanopore technology. The big advantage of that would be, though, however, is that this allows you methylation analysis. And methylation analysis, I haven't had time to talk about this, but it's an important, DNA methylation is an important part of early detection of cancer. And then we will, on the clinical work package, we will look at the rituximab response and toxicity. We have done quite a bit of work already around sustainability. Obviously, we've done a lot of training. There are master students and PhD students on the program. We have created now the additional site at the Central Pathology Public Health Laboratory in Uganda, in Kampala, to create liquid biopsy sequencing capacity in Uganda. We will support the master program in bioinformatics here at MUHAS, both in kind with training modules that are aiming specifically at cancer analysis, but then also more generally by supporting some of the senior staff. Then we are in the process of spinning out a MUHAS company that will essentially be a social enterprise to deliver DNA-based sequencing services for research and diagnostics. So this is a MUHAS initiative that will, hopefully in time deliver a self-sustainable, cost-recovering sequencing facility. So in all of this, the main challenge, apart from COVID, of course, and aggravated by COVID, but predating COVID, has been procurement and supply chains for libraries and consumables. So, and this is a big play to any of the biotech companies that are listening, is to make sure that they are doing their work on their side, because increasingly I'm beginning to think that the fact why there's not much human sequencing ongoing locally in or regionally in Africa is because of the supply chain problems, and actually not because of a lack of training or lack of know-how. So last not least, the main aim really for the sustainability program will be to introduce early multi-cancer detection from liquid biopsies, and that is, at the moment, it's a huge gap in oncology, because I think most people have realized that early detection of cancer will be the key to cure and the key to a financially sustainable cancer management system. And the sequencing of liquid biopsies holds this promise that we will be able to detect cancer more quickly by regular blood tests. So this is exactly the same technology that we've now been using for the Burkitt's lymphoma kids, and to roll this out across adults and multiple cancers will be the next big goal for us. I will stop here, but not without thanking the many people who've been involved on this program, in particular my wonderful staff in Oxford, and then also, of course, the sites in East Africa. And then clearly a big thanks to all the kids who have agreed to participate and donate blood for the study. Thank you.